Hey guys, what's happening? Welcome to Your Forest. I'm Matthew Christoph, and I got a very interesting one for you guys today regarding chronic wasting disease. For you that don't know what chronic wasting disease is, it is a disease that gets into the brains of the cervid family, so that's the deer family, so deer, elk, moose, stuff like that, and it causes a slow and steady decline, and they basically become emaciated and really thin and salivating, and they eventually die. It's always fatal, so far as we know. It's only gotten into Alberta and Saskatchewan in the last, I think, 20 years, or maybe under that. And anyways, I brought on Catherine Cullingham. She is the product manager for systems biology and molecular ecology of chronic wasting disease for the University of Alberta. So she's been doing a lot of research regarding CWD and its effects and the genetics around it, the spread, what it is, where it came from, all that kind of stuff. So we talk about everything from, where it came from, what it is, to management, potential management strategies. So, and why you should care, why it's something that you should watch out for and get your meat tested and all that kind of stuff. So, I hope you guys like this one. I really enjoyed it. She's a very knowledgeable person. She has a lot of great information. Infinite amount of stuff we could have talked about. Just tried to keep it to CWD and not get carried away on bison and brucellosis and other things. So, but it was good. It was really good. As far as sponsors go, Greenlink Forestry, couldn't do this without them. Provide me all kinds of opportunity and resources to do this. Also, the Forest Resource Improvement Association of Alberta, same thing. Lots of resources that without them, I couldn't justify doing this. So thanks a lot. Also, Damaged Timber. Damaged Timber is an apparel company out of Edmonton here, and they are trying to support environmental sciences through the sale of their clothing. So they're 10% of everything they sell goes towards a scholarship or bursary, whatever they're calling it. And they're going to give that away, I think, in the fall. Here's the first one's going to go out. And they have awesome stuff. Really awesome hats, toques, t-shirts, hoodies, all kinds of cool stuff. So check them out. At checkout, type in Your Forest 10 and you get 10% off on everything else, right? So if you guys want to support something like that, please feel free to check them out, damagedtimber.com. That's about it. So without any further messing around, here we go. We're jumping right into CWD with Catherine. What's your background? How did you get into CWD?
It started with my PhD. So I did a PhD at Trent University in Ontario.
Okay.
And I worked on raccoon rabies. So I call myself a molecular ecologist.
Okay.
So I use genes to understand wildlife populations.
Gotcha.
And so what I was doing is I was looking at how raccoons disperse across the landscape, and you can do that using their genes to understand where rabies is going to spread.
Right.
And it was kind of cool because, we could find that rabies wasn't likely to spread from New York into Ontario through Niagara Falls, but they would across the St. Lawrence, and that fit in with the disease itself. So that's what we saw with the disease, and so that's what we saw with the genetics. So it was kind of a cool
They're not
PhD.
like jumping a truck and coming across on a truck or anything.
No, but interestingly, they did 10 years later, and then there was incidents in Niagara region because they did jump on a truck. So it was kind of cool that the data fit with what actually
Makes
happened.
sense. Like those little buggers got thumbs and everything, right? Like
they're smart little creatures.
Yeah.
Yeah. So I did my PhD there, and then I was looking for a postdoc, and an opening came up working on chronic wasting disease in deer at the University of Alberta.
When was this?
This was, I don't want to date myself, 2008.
Okay.
Yeah.
So that's like right when we started to see it coming in, I think.
Yeah.
Right?
So it was a big project, and, the government of Alberta was involved, and the university was involved. Saskatchewan was involved. Mm-hmm. And so I was, again, doing the same sort of thing. So using the genetics of the animal to try and understand how it's going to spread. Right. Are there landscape barriers to spread? Because we can tell that from genes. We can tell if a river they don't cross because the signature's in the genes.
Just quickly explain that to me. Yeah, I'm not really sure.
Yeah. So basically, so let's just talk about humans because people can relate more to humans. So let's say we have a population of individuals and they're all blue-eyed. Okay. And we have a population of individuals who are all brown-eyed. Right. If we keep them separate, they stay blue and brown. Mm-hmm. But if we let them move between each other, we'll get a mixing of population. So those are kind of the signatures we look for in these genes of animals, right? We look for genes that are distributed across the landscape and are they all equally distributed, or
Right.
are they specific to different areas?
Okay. So, like, are you looking for specific genes that correlate with like CW?
Nope,
No,
these are
so it's
all
like-
neutral.
Okay,
These
gotcha.
just reflect the process of the population. So
Okay.
basically dispersal and something we call drift.
Oh, I see. Okay, I see. You okay? Yeah. So you're seeing where, like, how the genetics are flowing through the population.
Yeah, exactly.
And therefore, where you might see CWD be transmitted. Type
Yeah,
thing?
so
Okay.
basically if deer don't move across the North Saskatchewan River and we have, disease on one side versus the other, then that river would be a barrier to disease and it could be a good opportunity for management.
Gotcha. Okay.
Right? So unfortunately with deer, there are no barriers to movement.
Yeah.
So basically, I mean, it spreads kind of a diffuse pattern, right? So you've got your focus and then it moves out from there.
Right.
And so that's what the research I did found. One of the really neat things that we did find was that family groups were related to disease incidence. So the
Okay.
more related you were, the more likely you were going to share the disease.
Right.
And that
Not
also fits with the deer because they
Right.
form these female groups, right?
Right.
Where mom, daughter, grandma, they're all in these same social groups.
Right.
So that if you get disease there, you're going to have a kind of higher prevalence right in that little region. So
I'll just say, is that more to do with just proximity as opposed to genetic similarity? Or?
yeah,
Yeah.
for sure. Like it's proximity, it's interacting with the same environment.
Mm.
And
Yeah.
so, because there's the whole environmental transmission side of CWD.
We'll get
So
into that.
it's hanging out. So that's how I got into CWD originally.
Gotcha.
And then I did a 1.5 postdoc, and then I left it for a while to work on trees and bugs.
Yep.
And then once the trees and bugs funding ran out, I came back to CWD, and I'm now managing a Genome Canada, Genome Alberta funded project on chronic wasting disease.
Okay, so there is a lot of stuff being done for CWD.
Yeah, this is a new project. So it started almost 2 years ago. And so it's been a really great investment. We've got really great partners. We've got really great buy-in. So we've got, we're working with the government of Alberta, the government of Saskatchewan,
Right.
Even the governments of BC and Manitoba have been sending us samples because
Yeah.
the next province to be hit is probably going to be Manitoba.
Right. Just because of the lack of mountains in the way.
Yeah. And it's really close. Like in
Right.
Saskatchewan, incidence is basically right on the border
Right. So right now in Canada, it's just Alberta and Saskatchewan
in wild populations.
that we
Yeah.
know of. Yeah.
Yeah.
Okay. Okay. So I'm trying to figure out how to do this where we're not jumping around too much. Because it's going to get, right?
Yep.
So let's just talk about, I had Tom Habib on from
Yep.
BHA on, and he spoke quickly. Well, let's just quickly get into what, like, the prion diseases and how they spread
Yep.
and that kind of stuff.
Yeah.
So just what is CWD basically?
Yeah, it's a fascinating disease. So it's chronic wasting disease.
Mhm.
And it is spread, as you mentioned, by a prion. So it's a protein. It's called a proteinaceous infectious particle.
Okay.
So what happens, let me back up one step. It's similar to other diseases that people are more familiar with. So mad cow disease, Creutzfeldt-Jakob disease in humans.
And that comes
sheep's
from mad cow,
scrapie. They're all different.
Okay, they're all different. Okay, sorry.
They're all different. Yeah.
Sorry.
Yeah. No, actually, it's a great question because they each have different properties. So something like Creutzfeldt-Jakob or BSE, it's not infectious upon like just indirect transmission. Like it's not transmissible unless you eat infectious material, right?
So if a person ate like infectious like
meat
from a cow, they could
get
potentially
variant Creutzfeldt-Jakob.
Gotcha.
Yeah.
Okay.
Or a cow gets it from eating another cow's infected meat.
And that's how that happened too,
Yeah,
right? It was all
exactly.
farmers were feeding them
Yep.
dead cows basically. And
Yep.
now that's illegal. You're not allowed to do that anymore.
Yeah, don't do that. No,
Yeah.
basically don't feed any animal dead things. And there's other versions. There's mink encephalopathy, so it's a similar prion disease. And there's a brand new one. There's one in camels.
Oh no.
Yeah.
Good thing we don't have camels, I guess.
Yeah, yeah. And so one of the things about CWD, it is transmissible.
Right.
So you don't need to eat somebody else's brain. You can transmit it from individual to individual. So the prion in CWD is shed through their saliva, feces, urine, velvet,
Right.
dead carcass. It contaminates the environment. So it's a lot more dangerous in terms of the susceptibility of the population. So they're
Right.
more exposed to it. It's
Right.
not just these instances where you have to eat it. So it's transmissible.
Right.
Scrapie is also transmissible. So
The
there's
scrapie, that's in goats?
Sheep, goats, yeah.
Oh, okay.
So scrapie's been around for, we don't know how long, like hundreds, thousands of years.
Right.
There's been documents of sheep scrapie, so that's probably the progenitor of all of these sorts of things, the kind of ground zero.
Yeah.
We don't know exactly, but CWD is more similar to scrapie in that it is transmissible.
Gotcha. So when we say sheep and goats, just for people, that it's wild sheep and wild goats. Like
Nope.
bighorn sheep.
No,
and stuff,
they're not in. It's only in domestic.
Oh, okay.
Yeah, CWD is the only one in wild populations.
Oh, okay. See, for some reason, I thought that was in.
Yeah.
I thought that was in like wild, like billy goats and bighorn
if they
sheep.
were exposed to it, they would be susceptible based on their gene sequence. Because what this protein does, so it's a native protein in us. It's expressed in the brain.
Yeah.
It's called PRNP.
Okay.
And it causes a misfolding of that. So it's really neat in how it transmits. So this misfolded version propagates misfolding in the native version in an individual.
Okay. So basically, if you get this prion protein that's infected, is that the right
Infectious.
word? Infectious. And it'll whatever other proteins it comes into contact with, it'll basically like zombify it, turn it into one of them.
Zombify, yeah. And it has to somewhat match in sequence.
Okay.
So, and that's why we haven't seen rampant cases in humans of CWD, because it doesn't match well.
Gotcha.
And we don't know the exposure either. We
Right.
don't know how much exposure there is, right? Like
Okay.
if even with BSE, we had this variant Creutzfeldt-Jakob resulting from people eating BSE meat.
Yep. BSE
How
being
many
mad cow,
millions of pounds of mad cow meat was out there, and how many people were exposed to it with a
Right.
handful of cases? So I think there's an exposure thing. Like it's a chance the right strain of prion and the right human version resulted in successful conversion in the humans and causing that variant Creutzfeldt-Jakob.
Right.
But yeah, so that's
So
chronic
stars have
wasting
to align,
disease.
really have to align perfectly for it to. So not necessarily will you definitely get what's it called?
So it's variant
Creutzfeldt-
Creutzfeldt-Jakob
Creutzfeldt-Jakob.
if you get it from BSE.
Right. Okay.
Because we have a natural Creutzfeldt-Jakob in humans.
Oh, I see.
And it's more genetically determined than anything.
Okay.
So yeah.
So anyways, CWD.
Yeah. and then CWD infects a whole host of cervids.
Okay.
So we have moose, elk, mule deer, white-tailed deer, and caribou.
And caribou too.
Yeah. So
Yeesh.
caribou have been infected in Norway.
Yeah, I heard that.
Yeah.
And they.
And moose.
Okay. Yeah, there was that Norway thing was interesting. And I don't know if I want to talk to it just yet. You know what? Let's do it. Fine. Let's talk about Norway.
All right.
Because yeah, like I listened to there was the Meteor podcast with Stephen Ranella. Anyways, he does he brought on an expert from the U.S. Forest Service or whatever, and he was talking about the Norway situation, that they got in the caribou and that they went complete, like they just basically destroyed that population in that area.
Yep.
And they're making sure that nothing goes in there for X number of years, so that it does not go into the rest of the population.
Yep.
So it's that serious of a disease, or that it.
Yeah. Because there's no treatment.
Right.
There's no vaccine, and it's hard to detect. So it has an incubation period of almost 2 years.
Yeah.
So in those first 2 years, when there's no physical signs of the disease, so it's called chronic wasting disease because the animal eventually wastes away.
Right.
They get really emaciated and skinny and sickly. But before that period, there's no way to tell looking at that animal.
Right.
And the best way to tell is to find this, what we call protein-resistant protein.
Okay.
And you can't really get to it unless you sample the brain.
Which is killing them.
Yep. So
or less.
lethal sampling is the best way to do it, which is why the government of Alberta has a massive hunter surveillance program. So if you're a hunter in Alberta, you know that there's drop-off locations for your heads, voluntary and involuntary. If I was a hunter, I'd drop off the head regardless of where it was, just to be safe. But it takes weeks for the results to come back. Right. So if you're a hunter and you've got a deer cut up in your freezer, you have to wait months sometimes, or at least weeks, to get the results. So the project I'm working on is trying to change that process.
Gotcha. Okay. So many questions. Okay, I have to try and keep it in line. Okay, so it's passed through the saliva and stuff like that. So a lot of people talk about them like nose to nose contact. So obviously there's saliva on the deer's nose, and it's just if it has CWD and it's been in there long enough that it's showing up in the saliva, if they touch another deer, it's basically like a sure thing.
I mean, we don't know exactly, but we know it's transmissible from
Right.
individual to individual and also from the environment to individual.
Right.
So there's been, experimental farms, when they originally found CWD, they had, removed all the animals with disease and then put new animals on the landscape, and those animals got it. So it can bind to soil and remain infectious, basically.
Yeah, like Tom was saying that and I heard from another couple other sources too, that they don't know exactly how long it can last, but like if it's in like feces or something on the landscape or just in the environment somehow through saliva or whatever, that it could potentially last years or maybe even decades. We don't know.
Yeah.
And that it could be transmissible that entire time,
Yeah.
or infectious that entire time.
Yeah, that's true. And some of the work that's being done on this project is looking at different components of the soil. Because some of them might help to degrade it. Some of them might help bind it and make it more available. So there's a whole component of the project looking at the environment and how it interacts with the environment to get a better understanding. Because soils, I just learned this from soil scientists, they're really different across the landscape in that, from Norway to Canada. And so there might be some soils that are better for CWD in that they don't make it available. And there might be some that are worse that bind it and keep it there for 10 years.
Right.
So it's like just that area in itself of research is super interesting and a lot of unknowns.
Yeah, no kidding. Yeah, it just seems like it's only really been here for 10-ish, 15 years that we know of or whatever. And it's like, so the research is just kind of starting, right?
Yeah. So, I mean, the first records of it in deer was in captive facilities, research facilities. And I think it was in the 60s they were there, but it wasn't really documented and named till about the 80s.
Okay.
And I mean, our wild populations didn't have it in Alberta until 2006.
Right.
So it's pretty new.
Right. Yeah, it seems like a scary one because, like you were saying before too, that it's wasting disease, right? So they could get it, and it takes like 2 years for them to start showing symptoms or more, even, right? Normally,
like from what we know, observing animals in captive facilities and things like that, and tracking animals on the landscape, about 2 years is generally the incubation period.
Okay.
But let's throw another wrench into it. Some genotypes of the individual. So I was saying how the proteins need to kind of match. Mm-hmm. Well, some of the genotypes in the wild population show longer incubation times. Okay. So we don't know if they're slightly resistant to it and that's a good thing, or if they're just shedding for longer.
Right.
We don't know.
Oh, okay. I see.
So there's some really interesting interactions between the prion and the individual's genotype.
So when you say 2-year incubation, you mean 2 years till you start to see symptoms, or just 2 years till, like, say it's like I guess you don't really know, right? I guess you can't really say like, oh, the deer, like, here, we're giving this deer CWD. Let's watch and see how it progresses. So you don't really know, I guess. You just know it takes a while before you start seeing symptoms.
I mean, there has been some experiments where they've been able to do that. And it's generally by 2 years, they'll have succumbed in that 2-year period, generally.
Oh, they will have died in those years.
But I mean, it's harder to tell because, in a captive facility, a deer isn't going to be subject to all of the things in a natural deer is. Yeah. So it might have a much shorter incubation period in the wild because there's more stresses.
Yeah, for sure.
Or maybe they hate being in captivity and that's more stressful. I don't know.
Yeah, it's hard to say.
Right? They're a wild animal held in captivity. So there's probably going to be differences between what we see in the captive animals versus what we see in the wild. And it's really hard to do these captive animal experiments. Like Alberta doesn't have facilities for something like that, right? Yeah. Those facilities are really limited. In Canada, it's not really favorable. People like animals and they don't want to see them held in captivity and experimented on. So it's really difficult to get at some of these questions.
Yeah, no, I can see that.
Yeah.
So it is always fatal as far as we know, right?
Yep.
So it'll eventually, if you get it, you're dead. That's not you, but deer.
Yeah. I mean, there is some, there has been some correlations done basically with wild populations looking at the genotype and disease incidence, and where some genotypes don't seem to have any disease incidence. Yeah. And in the deer, generally what we see is there's a difference in the genotypes with the disease, but all of the genotypes seem to have the disease.
Okay. So it shows up differently, just the same as the flu shows up differently in people. It's just,
Exactly. There's an interaction between a genotype and the disease itself.
Right. Okay. Yeah. So you spoke quickly about it showed up in captive. So that's when they first noticed it was in captive deer.
Yeah.
I heard something that there was, I don't know if this is true or not. It might have just been like a rumor or something that they might have been doing experiments in that same captive facility regarding what's the one in sheep? Scrapie. Is that true?
Yep.
It was.
Yeah.
Huh.
So it's likely been a...
Right. We can't say for sure, but that's a pretty interesting
I mean,
correlation.
there's a really... I don't know all of this research because it's quite expensive.
Of course.
But there is a whole kind of camp that says it's from Scrapie because it's transmissible, like Scrapie is and such, and because this captive facility at one point had sheep.
Right.
So it is entirely possible.
That's crazy.
Yeah.
So so this might be not it's possible this is not a natural occurring thing. It's totally like you know what I mean, we don't, as far as we know, it didn't occur in nature before this event.
Right. We don't,
in quotations.
Yeah. Yeah, like it's been in sheep for thousands of years, I think.
Right.
So
but it never got into anything else until
more recently.
Right.
That we know of.
Exactly. Yeah.
Right?
That's the thing.
Right, because we're now looking for it.
Okay. So places like, if it was originally found in the States, so for example, the Norway population you were talking about, was there deer from the States that got brought over there? Or is that a totally different population, or disease?
That one we're still trying to sort out.
Oh, okay.
Yeah. Well, and that's the global CWD research across the world.
Right.
So, because it's in moose and reindeer there. So they're trying to see, because there's strains of prion, just like there's strains of virus.
Yeah.
And so those strains interact with these different genotypes differently.
Yeah.
And so they're trying to get at whether these strains are the same between the moose and the caribou, or if they're all different, and what they're more similar to, like, because there's elk strains, there's mule deer strains, there's white-tailed deer strains. So we don't know exactly how many strains there are, but slowly the labs are characterizing them.
Gotcha. But it seems like they're all converging from the same disease.
Yeah, they're all part of CWD.
Right. Okay. So there's just it gets into a mule deer and changes slightly, kind of thing, and something like that.
There's a bit of an evolution, we think.
Okay.
Yeah, like the research is showing that if it's going from an individual. So you've got two individuals, one with it, one without it. Those two individuals have a different genotype. Mm-hmm. When it goes to that other individual, it goes through kind of a selection process, I think. And then if it goes to the same genotype, it further selects. Yeah. And then it's a bit different.
It's okay. If anybody, it's funny. In a few episodes, I have the like that panging noise or whatever. Like, I don't know, I can't make it purposely, but there's nothing I can do about it. It's just kind of in there now. But whatever. I think I had one episode, the pine beetle one, where it happened all the time. It was like clang, 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 clang. I'm not a good enough editor to pull it out, but that's okay. Whatever. Anyways, okay. Where were we talking about Norway and stuff? So we don't know for sure how it got into Norway, but
we don't know.
We do know that it started in the States and it somehow got out through nose-to-nose contact or something, through a fence or something.
That's likely to get to the wild population.
Right.
And I mean, that's how it came into Saskatchewan. So it came to Saskatchewan first through farmed animal that was infected from the U.S.
Okay.
And then contact with the wild.
So like the farmed animal was like transported into...
from the U.S. to Canada.
Oh. So it didn't like walk across. It was actually transported. They just didn't know it had it. And then okay.
So this is why if there's a disease in that animal, we should just always test it.
Yeah, no kidding. Yeah, exactly. Okay. Okay, I'm trying to keep us on track here. So what is the disease occurrence in Canada?
Yeah, so it varies depending on where you are. So like in some areas of Saskatchewan, it's up to 30%.
Oh, really?
Yeah.
Wow.
So, and I mean, if you go to somewhere like Wisconsin, where it's been for a while, there are some areas where the males that are hunted, it's at 50%.
Wow.
Yeah. So there is a pattern. It seems to be, at least in Alberta and Saskatchewan and most other places, that it's more frequently in mule deer when they're together.
Okay.
Because, I mean, Wisconsin doesn't have mule deer, so it's all in whitetail deer. Mm-hmm. But when they're together, it seems to be more in the mule deer than the whitetail. And it seems to be more detected in males than females.
Interesting.
Yeah.
Why would that be?
We're still trying to figure that out.
Say any extrapolations on that?
Yeah. So there could be a behavioral thing where males come into more aggressive contact with each other. So there's a higher probability of transmitting.
I guess if they're rutting and stuff in the fall,
beating each other up, kind of thing, right?
Yeah. They're bleeding,
and maybe it's just a behavior thing where females are less gregarious in their environments.
Right.
And males are getting out and doing things they shouldn't be doing.
I guess the males, they're more
they're
prone
marking
to travel
territories,
more, right?
maybe. Yeah, so it's hard to know exactly, or maybe it's when the females get disease, they have a different behavior, so they hide,
Right.
and we don't detect them.
Right. So you just don't know yet. Yeah. Okay.
Yeah, I think a lot of it has to do with behavior, and I
Right.
think that's why mule deer have it more than whitetail. I think it's a behavior. I think mule deer are more likely to group and interact with each other than the whitetails, maybe.
Okay.
And maybe it started in mule deer, and their genotypes are just a little shifted, right?
Oh, okay.
They do share some of the same PRNP sequence, but there are some differences between the two species.
Gotcha. So when was it first discovered in like the bigger ungulates, like moose and elk and that? And where was that? Was that here? Do you know?
That's an excellent question. Elk has been definitely like Rocky Mountain elk, I think, was when it was first found in elk.
Gotcha.
Moose isn't very prevalent. Like Alberta
Okay.
only has a couple cases
okay.
across the whole time span, right?
Right.
So it's susceptible, but not, and that could be because it's not interacting with individuals that have it. So
Right.
that's why it's not getting it.
Mhm.
And they're more solitary creatures, so they're not interacting with each other as much. So
Right.
there's less spread.
Yeah, I could see it getting into elk. It could be a real problem. But
Yeah.
moose, it seems like it's not like they're walking around in herds or
Yeah.
anything.
And like I said, like that strain thing. So if Alberta has fewer strains that are elk friendly, then
Right.
then we see it more in the deer and less in the elk. But
For
then
sure.
if the strain evolves in 10 years, maybe we're going to see it more in the elk.
Man, what a scary disease.
It's scary and terrifying and really interesting all at the same time. Like
Yeah.
from a science perspective, it's just fascinating to work on.
Yeah. It's really weird. It just like reminds me of like a zombie outbreak.
Zombie
Somehow.
outbreak.
I just my mind keeps going to that for some reason. I don't know
Well,
why.
you know, these emaciated deer and they're drooling and
Yeah.
Yeah.
So crazy.
It doesn't help.
Okay, so getting into like handling like hunting them, right? Like
Mhm.
hunting deer and elk and moose that
Yep.
and testing for that and all that. As far as we know, I say that because obviously there's some like gray area in all of this. Has it ever been found like transmitted between like a cervid and a human being? No. Not yet.
No.
Okay.
No. And there's a whole whack of research that's just coming out on this.
Yeah.
So there's been some transmission studies in macaque monkeys.
Okay.
And there's 2 research groups. The one just sent out a paper just a couple weeks ago, and they say no.
Okay.
It doesn't go into macaques, which have similar PRNP to humans.
Right.
However, they kind of bracket that with there are different strains,
Mhm.
so could be the right strain at the right time.
Right.
And like I said, when they're in that evolving phase,
Mhm.
that's when they're most susceptible to a species transmission,
Right.
because there's more kind of variance basically of the prion, and so there's a selective. So if in that stage it's exposed to a human, it might.
Right.
I don't know.
So we don't
Right
know yet,
now, I would say no, but I would also say don't eat CWD tainted meat.
Yeah.
Test your deer, right?
Right.
Like, and the federal government now and the Alberta provincial government says we recommend you not eat CWD
Right.
positive meat.
Yeah.
It's
Just out of the risk
Yeah.
potential.
It's out of the risk.
Yeah.
Yeah. I mean, why would you chance that?
Well, so yeah, it's good that it hasn't happened yet, but I mean, it because like for example, BSE, like the mad cow, that can be transferred. Is there enough of a difference there that we don't like researchers don't think that it's going to develop that way, or is it like I mean I'm just talking like risk, like potential. Like, do you think is there anybody out there saying that like I see like a 10% chance that it could be transmissible into humans type thing, right?
I think right now people are thinking it's not.
It's not at
But
all.
then
Okay.
they still couch that with, but we don't know because we haven't really looked at all the strains. We don't even know all the strains.
Right, of course. So it's just yeah, the risk is too high. Don't because if it did potentially get into us, it could have potentially the same end result, right?
Fatal.
Fatal. Yeah. Okay.
Yeah.
So,
And I mean, like I said, I think it's like that chance and level of exposure.
yeah.
So we interestingly, I think there was a banquet. I think this is in New York, and this is a while ago, and it was catered with wild game, and I think some of that was tainted meat.
Yeah.
So all the attendees of that banquet are being followed
Really?
medically. And so it's a natural oops experiment.
Yeah.
But like I said, if it's 10, 20 people, maybe they don't have the right PRNP genotype. Maybe it wasn't the right strain. Maybe it's just a matter of time.
Right. Of course. So better safe than sorry, for sure.
For me, yeah, for sure.
So what's the because my understanding is that it's still kind of in the agriculture areas, like kind of in the southern part of Saskatchewan and Alberta. It hasn't really worked its way up into like the boreal, into the bush and stuff like that. Is that true? Or
yeah, it's I'd say how far. So I'm just looking at the map in my head.
Yeah,
and it's kind of like Edmonton and a little bit north of that. Like it's
creeping then, so it's getting close.
Yeah. It's getting close. I'd say it's on the edge of that.
So basically, okay. Because I do most of my hunting in Slave Lake area, and like I've never had any of my deer tested, or my moose or elk.
Because it's not quite that far,
right? But it might
west,
be right
that we know.
Right. Would they even take it if I wanted to get it tested?
There's a voluntary submission.
Oh, okay.
So they have freezer drop-offs. You just go on their website and they've got freezer drop-offs at like places like Cabela's and things like that.
Oh, okay.
So you can just drop your head off and let them know where it's from. Because then if it is positive, and those kinds of things are really helpful for them, because then that gives them kind of a better idea of where it's moving.
Oh, totally. Yeah. Yeah. Oh, what a crazy. So, okay. Yeah, the eating the meat thing. So is there, as far as like handling it and all that kind of stuff, like there's...
I'd say the risk is pretty minimal.
Pretty minimal, yeah. So it would just be an ingestion thing. If it does end up, it would have to be an ingestion thing.
And you have to like put it all out on the table. Like an animal that you've hunted looks really healthy. Yeah. If it looks emaciated, you're probably not going to hunt it. Yeah. And if you do shoot it, you're probably not going to eat it, right? Yeah. Nobody wants to eat a sick, dying deer for whatever reason. Yeah. So you're getting it at the point where it has CWD, but it's not, we don't know where it is kind of in the system. Right. Is it all through the meat? There's just so many unknowns.
It's just never ending. Hey, it's like, what about this? Maybe not. Yeah.
This is a great conversation because I don't have to actually say anything.
Yeah. You're not held to the fire. That's good. Okay. So, do you know what the regulations are around? Like, say I shoot a whitetail or a muley that's ends up being infected. Right. Do you know what the regulations are around that? Like, are we still required to like keep the meat? I'm guessing not, if the government suggests.
Yeah, so the government calls you.
Right.
They actually call you if you have a positive, and they say, you have a positive, we do not recommend you eat the meat. Right. And here's what you should do to dispose of it. Okay. It's still your choice. Right. They don't come and barge through your door and take away the meat. No. But they recommend not to eat it.
Gotcha.
And then I think they give you a tag for the next year.
That's what I was going to ask. Yeah. Do you get...
there are incentives.
Right. So they make sure that you're not losing out just because of some freak disease accident. Yeah. Okay. So you will get a tag for the next year, you said, you think?
Yeah.
Okay.
Yeah. No, I'm pretty sure you get a tag for the next year.
Okay.
Pretty positive. Especially, I mean, in the CWD area, they want people to take the deer out to try and reduce the level of the population, because that helps reduce spread.
Right. Okay. So as far as... have we seen, what have you seen as far as like barriers to the spread? Anything yet? You said there's not much, eh?
No. Mule deer, they go everywhere. So when the genetic study I did, I had a couple samples as well in like BC on the other side of the Rocky Mountains. Yeah. And for mule deer, it didn't matter. Like they just dispersed really well. So it's what we call isolation by distance. Deer closer together is more similar. Deer further away are more different, and it's linear. Right. Whitetail deer, the only structure there was on the other side of the Rocky Mountains. Gotcha. Right. Whitetails don't deal with mountains that well. No, so really. Yeah. The mountains are a barrier to whitetails, but they're not a barrier to mules.
Right. So it could. Yeah. That makes sense.
So the spread can still happen into whitetails on the other side.
Huh. So there's really. So we really don't know like how to slow this down. Like right now, we're just kind of watching it and making sure that we know where it is so that we can keep people from eating it. But there's no real management involved as far as like eradication or anything, right?
So when it first was coming into Alberta, there was an active eradication. Okay. So they would go in when they found a positive and basically take out all of the deer within kind of a range around them.
Because likely they have it, right?
It's a really effective disease control method in wildlife populations. Ontario does it for raccoon rabies. Right. It's very effective. However, not very popular among the communities, right? They see these deer just being sharp shot from the air. It's not something you want to see. However, all of the stats show that it was really effective.
That's the tough thing.
It's the most effective way to deal with the disease, right? And it's just not very popular because people think that they're taking out all the deer and there's not going to be any deer to hunt, and
Yeah.
and cruelty, all of those things, right?
I
There's
know.
your hunters, there's your wildlife, protectors. I get it. As a scientist, it's the most effective way.
Yeah. Well, that's just it, right? Like, I think that's the case in a lot of conservation sciences when it comes to, like, listen, this, it's
sucks, not favorable,
right? Like, it kind of seems shitty that we're just going to go out from like a helicopter and gun down a bunch of deer, but it's like, that is, if we can stop this disease from spreading that way, then it's not like you're going to eliminate, like, kill all the deer in the whole province, right? We're trying to, if you can get rid of a small population and that drastically helps the spread of it, then why wouldn't you do it? But like you said, people get involved, and what's the same thing with a lot of different hunting stuff, right? Like even the grizzly bear hunt in BC, right? Like, there's the science says that there should be a certain number of individuals removed from the landscape for population dynamics and safety and a bunch of other reasons. But like, they even came out and said that, you know what I mean? This is not a science-based decision. This is a public outcry decision, which sucks. But
it's frustrating.
But I guess that's a democracy, right? Like, it's all about public, right? So it's an interesting parallel, because you want people to be able to vote and make decisions based on what they think. But if those decisions are based in ignorance, then what the hell's the point? Yeah. Right.
Yeah.
But also, we're not going to stop people from voting in elections because of they don't understand the issue, right? Like, that's super messed up. Yeah.
And so, like, I think this is why the research is super important, because like the stuff that this project I'm involved with is doing is super cool stuff. Like, they're trying to find better ways to detect the disease in individuals, ways to detect it in the environment. Once it gets into that boreal region, there's a lot of remote communities, a lot of, First Nations communities that rely on deer. So maybe we can create a toolkit for them to be able to manage it themselves and detect where it is. Yeah. And then the other side is, there's a lot of people trying to develop vaccines or treatment options. Mm-hmm. So by looking at the whole genome, so that the project title, it's funded by Canada Genome Alberta, so it's got to have a genomic component. Yeah. So it's systems biology and molecular ecology of chronic wasting disease. So we're looking at, what RNA is expressed with the disease, what proteins are being expressed with the disease, and trying to better understand the body's response to the disease that can, feed into vaccination strategies or treatment strategies. So there's lots of really cool research going down right now. Yeah. So we just knowing where it is and how prevalent it is helpful, so we can kind of track it and maybe eventually contain it if we get some research breakthroughs. But research wheels move slowly because there's so many unknowns. Yeah. Well, that's just it, right? And it's such a new thing that, like, research takes time because you have to repeat the research. You have to, you got to test it. Like, we can test it on lab specimens, but then we need to find wild animals, and that's a lot harder to do because of everything to do with wild animals. Yeah. That's why I liked working on trees. Nobody cares if you took a branch off a tree. No, they really don't. Yeah. They don't care. No. You don't need permits for that. Yeah. You can grow them in a greenhouse and manipulate them. Nobody cares about that. They don't have feelings, right? They don't have feelings. Yeah. Maybe they do. There's an argument for that, but there is. We won't get into that right now. It's a whole nother different topic. Yeah. So, I mean, it's a really exciting place to be right now in terms of science and research and wildlife management, because there's so much going on, but really frustrating at the same time. Yeah. And that's the thing too, like, with as far as the fatality of it and the seriousness of it, I've talked to people who have said, they're like, well, it's not always fatal. It's like, well, what do you mean it's not always fatal? And they're like, well, like, I shot this one and it didn't have it, or it did have it and it didn't die from it. It's like, well, it was getting there, right? Like, it's chronic wasting, it takes time, right? So, yeah, of course, if it had CWD and it got hit by a truck, like, it didn't die from CWD, but it was gonna. Yeah. Yeah. But yeah, like, so the seriousness of this is pretty, serious. Yeah. Yeah. Like, so this has the potential to basically. It could really affect the whole kind of ecosystem, right? Like, deer are an important part of the ecosystem, and you take, you get a prevalence of 50%. So I'm wondering, have we done any research regarding the effect it'll have on the population as far as numbers? Because it takes time, it seems like it may not affect, like, progeny rates and that kind of stuff. And I don't think it will, because they're still able to reproduce. Right. And that's why, like, people are like, oh, maybe we'll get genotypes that evolve. And I'm like, it'll be hard to do, because the genotypes that aren't evolved to resistance, if there is some, they get to procreate in the population. Like it's not selective in that way, right? It's not taking them out right when they're babies. It waits, so they can be reproductively active even with it.
Yeah, and I guess there's probably, and it's not like you would want to find a resistant deer with the specific gene that's resistant to it and try to breed that into the population, because there might be some other aspect of that gene that is unfit for the population or something, right? Like you don't know.
Or that the prion evolves to that eventually.
Right. Hopefully.
Right.
Yeah.
The prion's.
You mean the other way?
Yeah, the prion's going to eventually evolve to that resistant version,
and I think that's take it over. You mean it won't be resistant anymore? Basically,
I think when the prion first comes in, it can only infect certain genotypes, and then as it gets in the population and more exposure happens, then the chances are it's going to. I mean, it's a 1 amino acid change, maybe 2.
Right.
It's not much.
Right.
And so I think there is. Even if we have some resistant genotypes, I don't think they're going to be resistant in the long run.
That makes sense. Yeah. Because the disease is evolving at a rate so much faster than the population, like the deer population is. So, like, what is? It's hard because it's not alive, right? It's just a protein. Right? So it's not,
and it's just folded wrong.
That's all it is. It's just folded wrong.
Yeah.
But
so it causes like plaques in the brain. It's really similar to things like Alzheimer's.
Yeah.
Because Alzheimer's is a protein misfolding as well, and it causes plaques in the brain. So there's lots of really cool research in protein misfolding in the brain and stuff. So this research can feed into human, and human can feed back. Like we can use some of the same techniques and learn things from each other, which is kind of cool.
Related. It's all neuronal kind of. Because there's something I read something that I guess I didn't get to that at the very beginning. We should have said that. So this protein, it causes neuronal death, right? Kills neurons and creates like kind of.
It creates holes in the brain.
And that's why it's supposed to be like spongiform.
Yeah, spongiform encephalopathy. Yeah, because it makes your brain, if you looked at your brain once you're dead, it's spongy. There's an old X-Files episode. And it's Creutzfeldt Jakob, and it was funny because when I watched it the first time, Scully had the slide on the microscope and she was looking at it. And as soon as I saw the slide, I'm like, that's spongiform encephalopathy.
Nailed it.
And then she said it, and everybody was like, whoa. Like, yeah, that's right.
You're like, yeah, I could be an X-Files. No big deal.
I could do that. Yeah. So it makes it look like a sponge.
So yeah, it's not like to the naked eye. Like if someone kills
no, it's like microscopic.
Right. So yeah. So it's not like you
but
can just
think of
see
these little microscopic holes in your brain.
It's going to cause problems.
It's going to cause
generally
problems.
Holes in the brain.
You need
not a good
your
thing.
Brain as a whole piece.
I'll make that as a quote for the podcast. Generally, holes in the brain, not a good thing.
Yeah.
Yeah. So I have so many questions regarding like strategy for management, right? So you were speaking earlier about potential vaccines.
Yeah.
Is that a thing we're working towards?
I mean, that's an area of research that people are doing.
Because that would be interesting. I mean, it's not like you'd have to go out and dart a bunch of deer, but you could probably, if you could do something that's like bait laden.
Well, that's the thing. Like the ideal would be with the rabies, how we've eradicated rabies in Canada. They use baits that they drop from an airplane.
Gotcha.
So they put the vaccine in something tasty.
Peanut butter.
I went baiting once for fox rabies. So I went baiting once and it's sweet smelling, like marshmallowy.
For sure.
And after being in a low flying plane that's really turbulent for a while, that sweet smell is not good.
A little queasy.
But yeah, you just you figure out what kind of density you need for baits to get the right exposure level, to get the right level of conversion in the population. Mm-hmm. But we're not there yet. Right. Yeah. That's the kind of ideal goal would be to have something like that. But I mean, if you had something that was just a shot, then at least you could help farms, like, deer farms. There's quite a few in Alberta and Saskatchewan, and they do get CWD. Yeah. And so if you can vaccinate those animals, then at least we know that supply, because that sometimes is gone into the food supply, is safe. So even if you had something that was just something you had to administer, there's at least some populations you can help. Okay. And then I think that would also be a proving ground for something like that.
Right. So there is a potential, like theoretically, we could make a vaccine eventually. Theoretically.
I mean, that's the ideal. They still haven't been able to make a vaccine for Alzheimer's though, right?
That's what I was wondering.
Treatment. Yeah. Treatment for Alzheimer's is really like some pharmaceutical companies have actually given up.
Oh,
Like
really?
they're just throwing in the towel. They're like, we've put too much money into this. It's not going anywhere.
Yeah.
Other companies are jumping on board.
Gotcha.
I think there's options, but who knows?
Yeah. I mean, I'm sure there's a lot of people who have been working on it. And
There's a lot of money that's gone into it, right? And they're still struggling with it. And we have a wild disease which doesn't see nearly the amount of financial funding as human disease,
No, of
right?
course,
So
yeah.
it's a big struggle. Like, what do you target your research on, and what aspects of it do you really delve into? And I think understanding more of the animal and how it's responding to the disease will help.
Yeah.
I think this whole like genomics approach and looking at the whole system, looking at the metabolomics, looking at proteins and things like that will really help us get at what's going on and what can we target.
Yeah. No, for sure. Yeah, it's hard to convince people that like, no, we should put this funding towards like deer brain problems, not like human brain. It's like, no, that's not going to happen.
Yeah.
It's like, I'm pro people, but
yeah. And I mean, all it would take is one human case.
Yeah.
Of a transfer.
Yeah,
and then, I mean, the funding, there is good support for CWD research, at least in the Western provinces. I think Ontario is quite aware of it because it's in New York. So I think Ontario or Manitoba could see it relatively soon.
I hope not, but yeah.
Yeah.
Yeah, that's a scary situation because everything I've read about it so far, it seems like they're like, it hasn't been there yet, and we don't think it will, but that doesn't mean it won't.
Yeah, totally.
Yeah, it's like there's still a potential there for it to, because like the replication rate of it is so fast. Right? Like how long does it take for the like a prion protein to
misfold a whole bunch more?
Yeah,
I don't think very long.
Yeah,
I don't know. I mean, if you compare it to a virus, like rabies, you're going to have similar incubation times. Rabies probably a little bit shorter,
right? But it's fast. Like it's evolving quickly, right?
Yeah.
So yeah.
Like there's multiple generations within an individual of misfolding and things like that.
Yeah. So every time it gets into one individual, it's changing one way or another, right? Yeah.
Yeah. I mean, the nice thing is that mule deer aren't very variable in their amino acid sequence.
Okay.
Like across, Alberta, there's only 2 or 3 different proteins.
Gotcha.
Which is nice. I think that's a good thing because then there's less opportunity for it to change.
Right.
Whereas white-tailed deer is a bit more variable, 4 or 5 that I've found so far and that other people have found. Elk is pretty boring, 2. Moose, pretty boring, 2 or 3.
Right.
So I think the fact that they aren't overly variable makes them all susceptible.
Right, of course.
But also keeps it kind of within those populations, hopefully.
Right. No, that makes sense.
And not evolving to others.
Yeah.
Because they're all relatively similar.
Yeah. Did I ask you already how many elk and moose were? I did ask you that.
Yeah,
okay.
we talked about it.
Yeah, because that's I wonder if it could get into so it's kind of stuck to the cervid family. We don't expect it to go into like bighorn sheep and stuff like that. Or
so that's one of the things that this project's doing is we're sequencing bighorn sheep in Canada as well as mountain goats, pronghorns. We're sequencing
pronghorns. I didn't even think about pronghorns. Yeah,
bison because bison are similar to cattle. So BSE is a
right.
So anything that's sympatric that could be at risk, we are sequencing those as well.
Okay.
And sequencing their PRNP just to try and get at how similar is it to these deer, and are there mutations that might make it less or more favorable to conversion.
Gotcha.
So that's another component of this project.
Because they are like pronghorns, for example, they are genetically very very different from the cervid family, right? Because they're different. That's a different family. Are they in the cervid family? I think they're different, right? Because they have horns, not antlers. They don't shed them.
Right.
Yeah.
Yeah, and we're actually just put them on the sequencer, so I should have data on those in a couple weeks.
Oh, okay, but so far it hasn't been found in pronghorn.
I don't think so.
Okay.
But there's something in the back of my head that says there might have been a pronghorn recently, so
okay.
I'm not going to say yay or nay to that.
Man, what a terrifying disease. Just the fact that we have no way of controlling it besides killing everything. Mm-hmm. So you said we were doing that and it got shut down because of public outcry.
Yeah. And now I don't think it would be effective because the environment's contaminated now.
Because now it's everywhere. Yeah. Right. Oh,
so
I see.
The animals that, you take all the animals out, but the environment's not, because it's been here for 10.
Yeah.
Over 10 years.
Right.
So the environment's now contaminated. So in a new area, I think that's much more effective.
Right.
Kind of maintaining it within an area.
Kind of like the Norway situation.
In the area it's in right now in Alberta and Saskatchewan, I don't think that would be effective because the environment's contaminated. So
That's
you'd
not-
have to like clear out all the deer and, I don't know, burn it.
Yeah.
Like,
Are they even susceptible to being burned? Because what I heard is that you need like 600 degrees Celsius to
they are
kill 'em.
pretty resistant. I think there was a researcher in the US, I think, who was just going to burn some landscapes, like not testing it, just burn some landscapes and just go for it.
Yeah.
So we'll see how that turns out.
Yeah, because that's
I think you need a sustained heat,
right.
and I don't know if a burn like that could sustain the heat you'd need for
Not a
as long.
flash burn. It would need to be like a deep organic burn that you burn basically all the organic layer off the top of the soil, basically, or
like
something.
you need something either super, super hot, like you said,
Yeah.
or hot and long.
Okay, so you couldn't even if it is in the meat, you can't even burn it out because it's just like I think it was 600 degrees was the number I saw to kill it, and like you're not getting your meat to 600 degrees. It'd be eating cardboard, not even cardboard, it'd be eating a rock.
Yeah, you could take that and turn it into a weapon. Probably
if there's anything left.
Yeah.
Yeah. Man. So currently is there any kind of strategy besides research?
I think research is the best
right
option right now. I mean, each province is going to deal with it differently because like Saskatchewan doesn't test the heads like Alberta does.
Right.
They don't have that kind of mandatory submission. So Alberta's really on the ball with this. Like they're keeping an eye on it. They're doing as much as they can. They're working with us for research. We talk to them regularly, like, okay, we're finding this. Is this helpful? What's helpful to you?
Right.
As we develop these research projects, we talk with them to make sure that we're going to be delivering them things that they can use.
Right.
And I think public perception is a really important part. So that's another component of this project where we talk about, how do people perceive the risk? Are taxpayers willing to put money in to this research to manage the risk? And some of them aren't and some of them are, right? If they're hunters, they're like, yes, I want to manage this risk.
Yeah.
Some people don't believe it's a risk. Some people think CWD is made up.
Really? Yeah, that's the other thing too. I've heard people say, why should I care about CWD when there's still a population, like the
and a lot of people are outside of it. They don't they don't even go outside. They hang out in the city. Why do they care about some deer in southern Alberta?
Yeah, no, it's tough. It's tough to get people to give a shit, unfortunately. But that was the other thing I was going to ask you is kind of just why should people care, right? If they're not there's obviously the potential for human. And like if it gets into people and it's transmissible,
well, then lots of people will care.
Yeah, then everybody's going to care.
Everybody's care. But I think the conservation groups care because they want to see functioning ecosystems, and diseases like this aren't a good part of them. They're not a natural part of them. They haven't been there for thousands of years. I think your hunters, they care.
Yeah.
And the educated people who kind of understand the disease, I care. Like I'm not a hunter. Well, I guess I'm a conservation person, so I fall into that, right?
Yeah.
So I think it's getting the people who are outside of those groups to be like, oh, this is an important part of the ecosystem. It supports biodiversity. And we want to maintain that. Like if all our cervids become sick, then that's an important part of that ecosystem. And then you can throw the caribou at them.
Yeah.
Caribou are susceptible. A lot of people care about caribou, even though they don't care about anything else.
Yeah, we found
they're iconic, right?
Oh, and
they're on our quarter, right? They're iconic and they're at risk.
Yeah.
And the populations in Alberta, if CWD comes in, that's a bad thing.
Yeah, no kidding.
We know they're susceptible. They're in Norway with it, and their PRNP sequence is really similar.
So far they haven't been in caribou in Alberta.
No.
Or
Saskatchewan. No.
Yeah. Okay.
And I think that it's a matter of time. They just have to.
Seems like it. Yeah. I mean, there's a lot less of them. There's only 3,000 caribou in Alberta or something. I don't know. Don't quote me on that.
Yeah,
there's not a lot.
But the scary thing is with Saskatchewan, you've got those big, like in northern Saskatchewan, you've got your big migratory herds that go up into the north.
Right.
And those are coming close. Their kind of southern range is coming close to where CWD is now.
That's the plains caribou. You mean?
Yeah.
Yeah. So
the tundra
also,
caribou.
So both are, I guess they're all cervids. They're all susceptible.
Yeah.
Man.
Yeah. And those caribou move. Like, it gets into those populations. You're talking thousands of kilometers, and then infecting that whole,
whole area.
Bad.
Yeah.
Bad.
That is horrible. What weird. So what, So as far as I don't know. My brain is just a mush right now. But it just seems really, really terrifying, and people need to start getting educated. And I even myself, I need to start getting my my animals tested because, like you said, right? If I do shoot something that ends up being infected up in the north, then that's Yeah, that's
that
a huge
helps. That helps us know where it's at.
Yeah, and potentially other people not
at risk that aren't testing. Right?
Yeah, exactly.
Because I mean, deer can move. Hundreds
do.
of kilometers. They don't always.
Yeah.
You know, they mostly stay within, but once in a while we get these crazy deer and we're like, I'm going to go on a walkabout.
Yeah.
And I don't
Especially
like
I always had that question regarding mule deer because it seems like as soon as like mule deer and whitetails don't really share much of a range, right? Like it seems to me like that when the whitetails kind of move in with development, the mule deer kind of move out. Right, so it seems like the mule deer might be more likely to move a little bit more like that, you know.
Yeah,
but
mule deer I think have a bit more of a migratory patterns to their movements than whitetail. They're more likely to do that.
Yeah,
but mule deer are also really friendly.
Yeah, I have a picture of one when I think I was like 8 or 10 years old up in Nordegg region. I was standing like feeding it. I think it was wild in the middle of the backcountry, like nothing. Like, they had probably never seen a person before, right? But yeah, it's yeah.
They don't spook the same way whitetails do. They're like, hey, what's going on? They're a little more curious.
Yeah, they're kind of dumb. I don't hunt them.
I like to call them curious.
People might take offense to me saying that. Yeah, I don't. Yeah, but yeah, it's a terrible. What about in like the Peace River region up there? They've been found in there?
No.
Okay. Because I know that's also like agriculture area. So
yeah, I don't think they're that far yet.
Okay, good. That's good.
Yeah, I haven't seen the 20. I haven't looked at the 2017-18 data.
Yeah.
From the province. I think it's on their website though.
Okay.
Yeah, the province is really great. They put all that data up so people know where things are at and where things are going. And, and they, you know, they're really great about informing their hunters and stuff. Like I really like how Alberta has been dealing with things and keeping things really open and accessible to hunters and keeping them educated.
So where would people go to get to find this information, like the drop-offs?
It's all on their website.
So which website?
That's just under the Alberta government website. So, so that's part of the Environment and Parks.
Environment Parks, it's in there. Okay. And then that'll have the maps of all the drop-off sites and probably some information, little factoids about it.
Yeah, they've got some fact sheets and everything.
Cool.
Yeah.
Yeah. No, it's a scary situation. Hopefully. So
I mean, I don't want people to run around and panic, but,
but
it's just something that people need to educate, especially if you're a hunter or you eat game meat in general.
That's almost exclusively what I eat is game meat. So it's a specific.
And I think a lot of Albertans do eat game meat. I think it's pretty common here.
Super common, I would argue, especially up in the north, right?
I mean, it's delicious. It's natural, and why not?
Yeah, exactly. It's the most sustainable way to do it, right? So, arguably.
Yeah, and you know, I eat elk burgers and things like that. They're delicious.
Yeah, it's a specific concern to myself because like I said, mostly all I eat is game meat. So yeah, I think I'm going to start doing that especially if it's just a free drop off and they're going to let you know and they're going to give you a new tag if it is, and then all the other things, right? So, do you have any like final thoughts? We're working up on an hour here already. So, any kind of final factoids or thoughts or interesting information you want to give out?
I think we've covered it. I think everybody's going to be digesting a lot.
Yeah, I think we have to grab a pen and a paper for this one. Yeah, I said that about the last remote sensing one too. I was like, hey, this is really specific. So like maybe like pause it, write stuff down if you really want to know, right? So,
and I mean, if you are concerned about CWD, talk to your policymakers. I think that's one of the important things is that if there is something that concerns you, you know, go to their pancake breakfast and say, hey, this is a concern to me. So then they know that there is public interest,
right?
And making sure that really cool research that we're doing that can help manage and understand this disease better, protect, you know, other animals, current populations, that sort of thing.
Yeah,
that there's a need for it and there's a want for it. I guess there's a want for it.
Yeah, that's the tough part because I imagine I find hunters and anglers and that kind of stuff, the people in those areas, they tend not to be super vocal about it because there is so much hate out there for that community, right? So people tend not to put themselves out there in that way. But yeah, it's important that we do, and just to try and education and all that kind of stuff, right? But yeah, it always comes back to education. It's one of those things. I remember I've said this before. It's in my environmental policy class in university. My prof came up to, I finished a presentation, right? And I said, well, and the solution to this problem is educate the public. And he just kind of looks, he's like, oh yeah. Don't you think that's the solution to literally everything? You're like, oh shit.
You know, people tuning into things like this helps educate them. Then they know more, and if they're interested, they can look up more. Yeah, right.
But it's a tough one, right? Because you have to try and educate the public on everything. So like for someone to take a specific interest in something like this, like I get how like if you're not a hunter, you don't live in the landscape, you live in downtown Edmonton or Calgary or something like that, and you don't, like, I could see how you have other concerns, right? But it's like, yeah.
You see people that are stricken with poverty, and
Yep.
you see people with cancer and all sorts of things, right?
Yeah. So there's a million different projects, but yeah, it's important that we look into all of them. Try and get everybody as interested as possible.
Just a little piece of the pie for this one, a little piece of pie for that one. Yeah. I mean, it's all important. It's all relevant. And
yeah, exactly.
Yeah.
Perfect. Well, awesome. Thanks for coming on. This was really, really good. Yeah.
All right.
That's it.
Okay.
All done. Thanks a lot for listening, guys. If you have any questions for myself or Kathy, please feel free to shoot me an email, yourforestpodcast@gmail.com, and I can get a hold of her, and we can get your questions answered for sure. Absolutely. So thanks a lot for listening. I appreciate you guys checking it out, and please feel free to rate and review and comment. And yeah, the more information I get, the better I can make this whole thing. So thanks a lot, guys. We'll see you next time.